be given by Matteo here. And so Matteo is a research staff member at IBM Research here in Ushika. He got his PhD in a joint program from ETH and IBM Research. And now he's working on deep learning, reinforcement learning, and I think mostly on two topics, cancer research and drug discovery, and the second one is what we will learn about today. So thank you.
Thanks. That's true, big topics, broad. So today I want to talk to you about the work that we are doing in the last 2 years, let's say, in our group, that are all around this topic of trying to use, to combine omics data together with information on chemical structure to be able to generate drugs using basically a bias given by the disease that we look at. So why drug discovery? Drug discovery is pretty interesting as a business for IBM for many reasons. The first one is that it's the largest R&D business that is out there, so around 20%, and this is, as of 2014, around 20% of the money spent in R&D business was spent for drug discovery. This accounts for 90% of the total. And also, there's a big delta in the sense that when we look at the drug discovery process, we realize that only very few drugs of the ones that are designed in the best early stages of the design and development make it to clinical trial or arrive in the end to the market. And this is a super expensive cost. So here there's a lot of margin where pharmaceutical companies could gain, but also patients in terms of like less expensive care or more access to treatment, right? So one of the main reasons, because drugs are very difficult to design, is that humans are all the same basically, but we respond to treatment in different ways, and this depends on a lot of factors. One is the genomic makeup, so our cells respond in different ways because they are just different in the different tissues that we have, and also under the metabolism point of view or interaction network point of view, they really just respond in different ways. A drug that triggers a specific process, maybe we work in a different way in other patients. So here, I just wanted to make clear why drugs tend to do not work. So medical chemists are very good at their job, but the problem is that it's a very challenging and difficult problem. And usually these two factors help us to put people in buckets or subpopulation that responds in a different way to treatment and therapy. So ideally, I mean, at least this is our idea, can be wrong or questionable, but what we would like to do is to use all this information in a holistic way. So we would like to put together the knowledge that we have about the interaction of proteins and transcripts and whatever is in the cells with the omic profiling of the patients, and to use also information from the fact that they belong to specific subpopulation to ideally design a compound. But first of all, a little bit of background on computer-aided drug design, and it's just one slide, so it's a very little background. I mean, there have been many ways to do drug design. It's not that we are just doing these things right now. So, I mean, there are tons of reviews on using genetic or evolutionary algorithms to design drugs and to optimize specific properties, and all these methods work quite well. And there are tons of machine learning methods that can be used to improve different steps of the process. For example, when you predict the sensitivity, you can use model to predict sensitivity of a drug before actually testing the drug in assays on cell length or whatever. So there are many steps where the process can be optimized and many ways to do it. And in the last 2-3 years, more or less, there has been like I don't know, an offspring of methods that are using deep reinforcement learning in different ways, and they are pretty successful to generate drugs following certain policies and optimizing specific properties. So, I mean, I'm just mentioning three, there are many, and they can even be better or worse than that. I just wanted to give you a brief overview. So there are models that are using an adversarial reinforcement learning scheme to basically optimize the moves that the model is doing in generating the structure of the chemical, or models that are simply looking at fragments and ways to combine them in a strategic way, or is one of the most recent and also one of the most successful. They are using sequence-to-sequence model just to generate drugs with given properties. And these models and all the models in this big family of models are super good because they are extremely effective in generating the compounds with desired properties, and they are very cheap in terms of data that you need to study, to use to train them, because you just need the chemical structure most of the time. So this is super good, but as you can see in this equation, all the parts regarding the omic nature, so the environment where the drugs act or the patient, is completely disregarded. So this is really the main issue that we have here. So what we would like to do, and what we are trying to do, and hopefully we might be able to do one day, is to use this information and generate drugs given a biomolecular profile. It can represent a cell line, a patient, an average profile for a disease. I mean, you can really think in a pretty generic way about this. So the first thing is that we wanted to formulate the problem in a reinforcement learning framework, and basically we have a state, that is the biomolecular profile. We have our actor that is trying to generate drugs given this biomolecular profile, and then we would like to have a critique that is basically considering the state and the candidate compounds to give basically a reward that is proportional to the property that we want to optimize. So basically, this is a very basic actor critique framework. You can resolve it in many different ways, A to C, A to C, reinforce. So there are many algorithms to do that. So the main interesting part of what we did is what we did with the critique and with the actor. Because these are the two things that are more difficult to adapt in this setting of computational systems biology. So as far as concerned for the critique, what we did is that we developed a deep learning architecture that is using multiple levels of information. So it's called Pacman, and we are using compound structure, prior knowledge and interaction between proteins, let's say, transcripts, but gene expression. I mean, I don't want to give much details on this because maybe there are not many biologists, but you can imagine this as how drug targets then interact with other proteins in the cell, and we use basically gene expression data. So what we do here is that we put together these three levels of information and we predict the efficacy of a drug. And to account for the chemical compounds representation, we use SMILE strings, so we use a string representation. It's a formal language where you have a set of rules to describe a molecule. And for the gene expression, we basically use a network propagation scheme to pre-select some genes, because we want to reduce a bit. We don't want to use all the genome, but we use around 3,000, so let's say 1/5 of the genome. No, 1/10 of the genome. So basically we use this network propagation to inject prior information about drug action, and we select a subset of gene expression values. And what we design is a multimodal encoder. So we start with very basic encoding of the SMILES. We tokenize on atoms and bonds. We then designed a self-attention mechanism to help the model to focus on specific genes when they're inputted in the network. And we combine these two different types of encoder in another attention head, basically. So in this way we have an attention level of the genes, so we select genes where the model has to focus on to predict the efficacy, and then we use the genes as a context to then have an attention on the molecule, on the drug. And then we wrap everything again in another set of multi-head attention. So this is basically inspired by transformer architectures, where we will have on the chemical compound different views, basically, different point of view. At the atom level, few atoms, more atoms. And basically this is what we do. And then we predict with just a few depth layers of NLP. Pacman works pretty well. I mean, we compare with some published methods, some self-designed things. I mean, we didn't come up with this architecture as our first thing, so we tried many things to get something better, but from publicly available data it performed really well. And the next thing of this method is that you can really then get interpretable signatures for your drugs. For example, you can really see where the model was focusing when the prediction was done. This can be very important for medical chemistry to look where the model was focusing and what was important for the prediction. And on the gene level, we really get relevant genes for the prediction. So this is just a leukemia case where we actually highlight genes that are related to leukemia. But I don't want to, we can discuss about Pacman later during the apero. I want to keep the focus on the reinforcement learning. So basically now we have a critique, and the critique is Pacman. And we have to solve the problem of having a good actor. And this stuff, because we don't have so many match data of compounds that are suitable for a specific cell line. So it's not something that is really out there, especially if you want to make sure that you are not using the same data here and here to maybe bias the model to have a super good predictor on this specific small data set. So on this side, what we decided to do is, why don't we just warm up and pre-train? So on one hand, we decided to find a way to encode the gene expression information using publicly available data, and on the other end, we decided to warm up the decoder, the generator of molecules, just using publicly available data. So for the gene expression, what we ended up using is a DAE. The gene expression are basically floating point values with just dense layers. We get a latent space out. And this was pre-trained on human samples. So before, up to that point, we just used cell lines. Here we are using human samples, anonymized from different cancer types. So we have a good encoder for the gene expression data. For the smiles, we did something very similar. Actually, we want to acknowledge two papers that really inspired our work. We stole ideas from both of them and we combined them. So one is a paper that was published last year where they're basically suggesting to use DAE to do molecular design because they are a bit easier to train. I mean, they have very good set of motivation, and then you have this smooth latent space where you can sample compounds along a direction, and you really see this morphing that a compound is transformed in a direction. This was very good. I mean, initial time really. And the other is the paper I cited before, where they use sequence-to-sequence model. Here we just stole the idea of using the stacked GRU cells. And stacked GRU are basically a GRU cells or an RNA cell where you keep also a sense of memory. And this is super useful with mice because you basically have to close parentheses, put numbers back and forth, so you have to remember a lot of steps later in the sequence when you have to close a parenthesis in order to have a valid representation. And this type of cells helps a lot. So we basically combine these type of RNA cells with the RNA architecture. And what we did is that we went to Canva, so no bias towards anti-cancer compounds, and we just trained a model to generate molecules. Basically, we trained it on all, I think around, not all, but 2 million of compounds. So now we have these two blocks, but still, they are disconnected. So this is still quite of a problem. And basically we have two things that we don't need. There are the encoder. I mean, we might use this as a primer, okay? But we basically don't need this information and this information. So what we started to think was, how can we put together this latent space? There are many ways you can put an MLP there, that you train all in the reinforcement learning phase. You can combine them with another math product or just with a sum. So the experiments that I will show you today are with a sum. We obtain analogous results with an MLP. So basically, we train the two architectures in order to guarantee the same latent space, so we don't have many calculations to do here. And what we do is that we provide the state, encode it, sum it. If we don't have a primer, here we are just summing to the origin, decoding, and then providing the candidate compound and go back with the reward. And then we update using the policy gradients all these weights. So we froze Pacman, and we update these weights. At the beginning of the training, this is quite unstable because we are basically sampling from a latent space that is completely different, but after a few iterations, the model really starts to get used to the new input and starts to generate compounds, as we will see in a second. So basically what we obtain this way is a way to provide the gene expression and get out a compound. And as I said before, you can even prime it. So if you use this information that is going into the latent space close to these compounds, you can even try to bias the generation of the compounds towards something that is somehow known or that you like because it's your favorite drug or your favorite chemical structure. So we applied this algorithm to 4 different cancer types. I will just show you 2. Then if you want to see other plots, we can do it, but it's a bit redundant, I think. So here I'm sorry because you really don't see this line, but basically what we are doing here is that we are showing the distribution of the IC50 values, so the efficacy of the compounds that we sample, This is in logarithmic scale, and usually 0 is rule of the thumb effective compound. That's simplified. So the more you go left, the better it is. So this is the generator before the reinforcement learning. So we see that the mode is clearly true, not many effective compounds, only 4%. Once we do the training to biases, we are actually able to optimize this compound. So 30% of this compound are now effective, and we are still keeping a good smile validity. So what we have at this point, once we train this model for our group of training cell lines, is that then we can go to test cell lines and check whether these things make sense, or sample drugs for a specific subtype of a disease, or, as I said before, for an average profile. You take all the cell lines of ER2 positive breast cancer that you have, and you try to sample molecules from this space. So this is quite a cool thing. To better compare it, because it's difficult, you should synthesize the compound at this point and then try it on a validation set. So we are working on it right now, we are still not there yet. But what we do is that we basically decided to look at other drugs metrics. I will briefly explain what they are. This is the drug likeness, so it's basically how beautiful a drug is. You can see it in this way. So, and there are models to estimate this number given a compound. The solubility, there's an optimal range, it is usually in this range, so we are quite far from that. And the synthetic accessibility is the number from 1 to 10 that tells you how easy it is to synthesize a compound. Because when you generate a compound, it's super effective looking at your model, but then it's impossible to get an hold on it and generate it in the lab. So basically, drugs usually are around 4 to 1, I think. But we are almost there. Keep in mind that here we didn't optimize for this property, right? So we were only optimizing for the efficacy, as I said before. So this was just as a sanity check. I mean, if this number would have been 0 all the time, I would have been a bit worried. And this was an FDA-approved drug. So this is the range of FDA-approved drugs. Okay? And we did the same experiment with lung cancer, breast cancer, and prostate. Here I'm showing just the results for lung, where you can clearly see that we are able really to move the peak of the distribution. So it can really be, you can have different level of effectiveness, and you can really check all these statistics and generate a lot of drugs that are actually novel. I didn't mention it before, but when you even compare it with known stuff, you see that we are actually different from what is usually contained in libraries. So it's also interesting under an IP point of view. And as I said before, I mean, here we are quite good at in terms of synthesis, but then we don't optimize this property. And just to, again, keep the enthusiasm low, I don't know why I did this to our model, but basically here we are showing through libraries of publicly available compounds, and we measure these properties that we don't optimize, just to understand how far we are from something that could match public libraries. So in terms of solubility, we clearly see that our method has longer tails. Usually following the Pischier rules, you should be in this range. We generate pretty tricky compounds to synthesize, because usually drugs are in this range. And for drug likeness, we have a decent overlap, but we have still to work on it. So basically, the take-home message of this presentation is that we are actually able to generate compounds conditioning on a biomolecular profile. So it was something that we really like to see and observe. But we have work to do. So the next step is obviously to add in the reward components that are measuring these properties, if we really care about these properties and we can measure them. Ideally, it would be nice to have toxicity, but predicting toxicity is super tough. These are more low-hanging fruits, we'll say. It would be good to have protein information, because when medical chemists design a drug, usually they have in mind a target. You have these bioinformaticians or biologists that are working in the lab, and they are saying, okay, this is a target makes sense, this pathway is what we want to do, so go for this protein. And this is something that we don't have here, and there are many ways to do it. I mean, we have plenty of information, not on 3D structure, but on 2D, not 3D representation, amino acid sequence information. So we might, for example, train unsupervised language models to get embeddings and encodings for proteins, and do the same exercise that we did with the gene expression. And then there are many other things. I mean, a lot of papers are using graphs to do this type of work. We didn't use graphs for historical reasons, in the sense that when we started to work on Pacman, it it was more handy to work on smiles because we could augment the data. So, but this is something that we should really look into. And the other nice thing is really that genes and gene expression values are not just plain vectors. I mean, you have a topology there, and we know a lot of interactions. So it would be cool to see also the gene expression as a graph and use graph neural networks to do the encoding, so that we are really trying to use all the information at the same time. And with this, I thank you for listening to this. I think you followed it. I am happy because biology can be quite boring. And I thank all the collaborators, Ali. We started to work on this together. Joris is a data scientist, he's not here, he will be punished later. And Marielle is our group leader. So if you have any questions, I'm happy to take them. And I hope I can